I am Donald Hodges, and I would like to welcome you to this Talking Tox webinar hosted by the American College of Toxicology and sponsored by the CBSET and CILcare partnership to advance auditory therapies. Today's webinar is best practices for the conduct of preclinical ototoxicity evaluation. Next slide. Go to the next. Today's webinar will discuss the value of ototoxicity assessment and some of the challenges. It will cover why it is important to front-load ototoxicity assessments. How would you do a GLP or non-GLP ototoxicity assessment? What are the current regulatory guidelines and what is expected? I would now like to introduce today's speakers. Aurore Marie. Aurore heads up the global R&D operations at CILcare, working between Boston and France. She manages and monitors CILcare's French and US-based studies and ensures the highest level of excellence with the technology platform. She also served as principal investigator on GLP ototoxicity and general toxicity studies over the last 3 years. Our next speaker is Dr. John Keating. Dr. John Keating is a diplomate of the American College of Veterinary Pathology with 12 years of experience in contract research and preclinical safety evaluations, focusing on medical and combination devices, as well as toxicology studies. Prior to that, he worked for 7 years in academia and has a broad experience in gross and microscopic pathology, as well as development, execution, presentation, and regulatory navigation of preclinical studies. Our last speaker will be Dr. Christopher Toscano. Dr. Toscano received a PhD in toxicology from the Department of Environmental Health Sciences in the Bloomberg School of Public Health at the Johns Hopkins University in Baltimore, and served as a postdoctoral fellow in the Brain Physiology and Metabolism section of the National Institutes of Aging at the National Institutes of Health from 2004 to 2008. Dr. Toscano became a diplomate of the American Board of Toxicology in 2008, just prior to joining the U.S. Food and Drug Administration as a non-clinical reviewer. He is currently serving in the Division of Pharmacology and Toxicology for Neuroscience, where he is involved in the review of non-clinical studies conducted to support the development of neurology drugs. With that, I will ask Aurora to take charge.
Thank you so much, Donald. So first of all, let's have a look at the definition of ototoxicity. Here is the one given by the Ototoxicity Committee from the Department of Defense. It's defined as a damage to the inner ear, targeting cochlear and vestibular structures and sensory functions due to the exposure to certain pharmaceuticals, chemicals, or ionizing radiations. In this presentation, we will be interested in drugs with potential ototoxicity, causing hearing loss, tinnitus, or vertigo. More than 2,000 marketing drugs are ototoxic, with some leading to irreversible hearing loss. The most important ototoxic drugs are platinum-based chemotherapeutics, with up to 60% of the children receiving this kind of chemotherapy left with severe hearing loss and a very poor quality of life. Certain antibiotics, which are widely used like aminoglycosides, can cause hearing impairment to nearly half of the patients. There are many other drugs currently investigated, including drugs currently investigated for COVID-19, anti-malarial drugs, and also erectile dysfunction drugs. This is why it is crucial to take into account the auditory assessment in the drug development. So which drugs are currently concerned for ototoxicity screening? The first category is the new chemical entity, NCE, that has the ability to penetrate an intact tympanic membrane. For example, some antibiotics delivered by otic routes for otitis treatment. The second category is the NCE expected to reach the middle or the inner ear. This includes medication to treat ear infections with a perforated eardrum, for example. And the third category is the NCE, which are structurally similar to any other known ototoxic drugs and administered by systemic route. So the anatomy of the ear is very complex. The ear is composed of 3 compartments. The outer ear and the middle ear are separated by the eardrum and ensures that the transfer of the sound is transmitted to the inner ear. Then the inner ear contains 2 organs. A tiny snail-shaped organ named the cochlea is responsible for hearing, and the vestibula is responsible for the balance. The cochlea contains hair cells which transform a stimulus into a nervous message, which will then be sent to the brain. Drug ototoxicity can damage hair cells, auditory nerve, and sometimes even the vestibular system. Research and development has made tremendous progress over the last decades. Models and testing protocols have been set up and validated for both screening phases and in vivo assessment. You can consider different screening assays. There is the in vitro assay on otic cell lines from rodents. This allows an assessment at molecular and cellular level. An ex vivo assay on cochlear explants from neonatal rodents. This assay can provide data on various cellular cell types. And an in vivo assay in zebrafish. This model has morphological and functional similarities to the mammalian hair cells. And then there are the in vivo models available in rodents and non-rodent species for both non-GLP and GLP studies. Let's dive deeper into the early phase in vitro screening. So this in vitro assay has been developed by Professor Kalinek with HEI OC1 otic cells. These cells are derived from mouse cochlea, and they express specific markers including cochlear sensory hair cells and supporting cells. Different assays can be carried out on those cells, such as the cell viability, cell toxicity, and also caspase activation or ROS production to study the mechanism of action of drug candidates. Here is an example of the results of an ototoxicity assay with cisplatin. You can see on the images some untreated cells, cells treated with the vehicle, and cells treated with cisplatin. With the latter, cells are far less numerous. The graphic on the right represents 2 different assays to assess cell viability with cell quantification and cell metabolism. Cisplatin induced significant cell deaths with a dose response. Another relevant ototoxicity screening assay is the cochlear explant. The study starts by harvesting cochlea from postnatal mice. Then the organ of Cordy is removed from the cochlea and cultured in petri dishes for 24 hours. After that, explants are treated with ototoxic agents such as the gentamicin for 16 hours, then fixed, stained with anti-myosin 7A antibody to visualize hair cells. They are then the images are acquired and analyzed, and extra readouts may also be included. The main advantage of using explant is that the work is done on preserved cochlear 3D structures and allows an assessment of various cochlear types. However, it requires a rare expertise and should be conducted by scientists with years of experience. Of course, it does not include any functional tests, and observation is limited to a cellular level. Here is an example of results from an Australian research team using neomycin at different concentration. The 2 images represent immunolabeling of cells after 20 hours in culture with neomycin 100 micromolar and 1 millimolar. Changes in cellular morphology and DNA damage indicative of apoptosis were detected using the 2-nail assay. DAPI staining in blue was used to stain all cell nuclei, while myosin 7A in red was used to stain the hair cells. The graphic on the right shows the inner and outer hair cells quantification. 3 days after neomycin treatment resulted in an almost complete loss of outer hair cells and 2/3 of the inner hair cells. The third and last screening assay is the zebrafish model. The species spontaneously regenerates its neuromast hair cells, which are quite similar in structure and function to the mammal hair cells. Drug candidates are administered in zebrafish embryos, and after incubation, specific staining is performed, and the neuromass counting is done with an IC50 calculation. You can see on the images the neuromass in the controlled zebrafish, whereas most neuromass disappear in the one treated with high dose of cisplatin. The graphic on the right shows the quantification of neuromass with different ototoxic antibiotics such as neomycin, kanamycin, and also gentamicin. Let's now move on to the in vivo models and their functional readouts. Auditory brainstem responses, known as ABR, are the golden standard functional measure to assess hearing thresholds. ABRs are non-invasive and translational, and notably used for screening newborns. They are electric potential recording from scalp electrodes following a sound, and they represent the activity of the inner ear cells and the auditory neurons. The waveforms recorded in the ABR arise from the auditory nerve and brainstem structure. Each wave arises from a single anatomical site, as represented on the figure on the right. Here are examples of results of ABRs in rats after administration of cisplatin. At baseline, represented by the brown line, the animals display normal value of ABR threshold, that is to say below 40 decibel. 3 days after cisplatin administration, this is the orange line, there is a significant increase of ABR threshold. This indicates the presence of hearing loss with a 40 decibel ABR threshold shift that would be considered as a disabling hearing loss in human. DPOAE is a complementary measure of the ABR to assess the auditory function. There are acoustic responses produced by the cochlea, which reflect the functionality of outer ear cells. The graph represents the DPOAE amplitude in rats 3 days after cisplatin administration. At baseline in brown, the animal displayed a classical pattern of DPOAE amplitude. But 3 days after cisplatin administration, with the orange line, there is a significant decrease in DPOAE amplitude, which demonstrates hearing impairment. Usually, ototoxic drugs damage primarily the outer ear cells in the basal turn of the cochlea, resulting in a high-frequency hearing loss, as we can see it here. And finally, the middle ear assessment, performed post-mortem, is allowed to observe the integrity and appearance of the eardrum and the ossicles after the administration of a compound locally, for example. A normal eardrum is expected to be transparent with no apparent vessels. And we can also assess the presence or absence of formulation residues after the administration.
Thank you, Aurore. Again, thank you for your talk. Please submit any questions by chat, and the speakers will answer them at the end of the webinar in a discussion, in a panel format. Our next speaker is Dr. John Keating. Please take control.
Great, thanks, Donald. So my goal here is going to be to present a brief overview of the pathology end of ototoxicity studies, just to give a general idea of what goes on behind the curtain. And as with any safety study, the more one understands about the basic pathology thought processes and lab processes, and there's no magic involved in either, the better off you are both preparing for study design and getting the most out of your results. So pathology assessment of any tissue in the body can have its own set of challenges, and those involving the ear are some of the most complicated. So it's critical that the pathology team is experienced in the procedures to be performed and also involved early on in study planning, working out specific study elements, doing any method development, etc. And these specific challenges of otic studies are on account of the technical complexity of the endpoints, but also just because of the inherent anatomy, and where you have very delicate and sparse tissue, and much of it being neurosensory, arranged in a complicated microanatomy and all encased within a bony shell. So it's a series of fun. And so harvesting the organ itself is really just the first challenge, and necropsy personnel should be experienced in extraction of otic structures. and obviously need to take care not to disrupt or fracture tissues during extraction. And they also need to be skilled in any additional work that's needed at the time of necropsy, such as gross assessments, sampling of any specific tissue or compartments, making adaptations to ensure a distribution of fixative, and so on. And then decalcification has to then be performed to dissolve the protective bone encasing the target tissue. And this is regardless of whether the endpoint is sectioning the tissue to generate slides or to microdissect it out. And EDTA solutions are the basis for this process, and they allow for digestion of the outer bony tissue, importantly while preserving histologically and anatomically the soft tissue contained within. And the process has to be carefully monitored, and particularly if larger species are involved, so that samples are both thoroughly decaled and promptly processed. And then additionally, for evaluations such as cytocochleogram or scanning electron microscopy, microdissection may be needed, or in those cases is needed, to expose and extract the organ of Corti, and manual trimming of cochlea under dissecting microscope in order to generate intact and consistent samples. It's a very meticulous and time-consuming task, and it's one that requires some of the most training and experience. Okay, so once tissue is in hand and ready to go, routine histology and immunostaining are certainly essential for analyzing tissues and responses to any interventions, and that's generally performed via semi-quantitative assays and the typical 0 to 4 scoring matrices. But cytocochleogram, shown here, is what allows quantification of sensory cells, and it's how you count the inner and outer hair cells in order to provide numerical data. And it's also important that this cell counting can be performed on specific areas of the cochlea, such as the apical or middle or basal regions, but also at specific target frequencies, which is done utilizing species-specific cochlear frequency maps. And then being able to demonstrate hair cells numbers can help in determining whether a difference is correlated with hair cell loss as opposed to some other functional issue. And then once numbers are generated, then they could be compared to in vivo endpoints such as ABR or emissions testing, and then also to histologic findings, such as in the contralateral cochlea in instances where right and left ears are designated for separate histologic and cytocochleogram endpoints. And this whole process of cytocochleogram is one of the most specialized pathology analysis that's routinely performed for ototoxicity studies. And it's a critical endpoint. But because of the multiple complicated processes that go into generating cytocochleograms, just keep in mind that this analysis can be performed only at a relatively small number of specialized facilities. And here, just to show you specifically some of the steps of cytocochleogram, here at top left shows a low magnification image of a cochlear segment that's been dissected out after decalcification and has undergone fluorescent staining with myosin 7A to specifically light up hair cells, and it has also been hole mounted flat onto a slide. The bottom image shows a slightly higher magnification view of the same tissue, but also one in which the cochlear image has been straightened out to allow automated counting. And this also shows a target zone between the yellow lines in which specific analysis can be performed, and in this case in the 32 kilohertz region. And then in the top right shows the same region of interest at even higher mag, with each fluorescent positive inner hair cell indicated by a blue dot. So one's able to capture numerical data, including the raw number of hair cells per unit length and the percent of hair cells present, by comparing the hair cell count against the total potential hair cell count, i.e. taking into account any empty spaces. And in comparison to cytocochrography, I think that histopathology processes are generally much more widely understood because they're fundamentally the same as occur with any other tissue. Getting otic sections to slide can be tricky, but it's a fairly straightforward process for experienced histopath lab. And likewise, skilled pathologists can generally get very comfortable with routine assessments on this tissue. And the parameters are typical of pathology assessment in really any organ, and they include things like necrosis or cell loss, fibrosis, presence of inflammatory cells and what type, presence of any exudate. And then specifically regards with the cochlea, the assessment should include and can focus on various cochlear structures. And in this image, we see a longitudinal midline, or what we call a mid-moleolar section through a cochlea, showing the very typical Christmas tree or ice cream cone-like appearance, with the center of the cochlea here, the turns proceeding around the outside. And this enables us to look at the sensory area so that we could see the sensory regions of the organ of Corti, spiral ganglion neurons in the center, the bony structures adjacent to it, the lateral wall, stria vascularis, etc. And those should also be those should all be evaluated as part of a cochlear study. And in this image, additionally, this study involved chronic indwelling of an intracochlear device. And in this case, one would need to look at direct device responses. Here's a device tract with some blood in it, a little bit of fibrosis around the periphery, and some inflammation associated with the device. And then one should also look at both local effects, obviously on sensory and other structures within the cochlea and the middle ear, and can also look at more distant effects. And histopathology is assessment is predominantly based on H&E staining, and depending on the study needs in particular instances, we could obviously look at other stains regarding fibrosis, highlighting specific cell types and so on. And this is just a reminder that depending on specific study needs also, then assessment could focus on the cochlea or other ear components, and cutting of multiple step sections across the available otic tissue, which is generally embedded whole and intact in a large paraffin block or a resin block, is basically always warranted. And that's whether to get a clearer view across the cochlea from edge to modiolus through the opposite edge, or to show different otic structures, such as in this case, vestibular organs, middle ear, or one might need a different plane such as this, in which the cochlea on the left, which is nicely demonstrated there. This is not, so this is a very peripheral section of the cochlea, not useful for cochlea, but what it does show very nicely is the end of the external acoustic meatus, the tympanic membrane, ossicles, and middle ear. So this generation of multiple sections, it adds a little bit to cost and effort and complexity in otic studies, but it can be really critical whether you're looking at the cochlea or other structures. So I hope that gives you a little look under the hood as to our thoughts and processes from the typical histology lab and pathologist perspective. And I'll turn it over to Dr. Toscano.
All right. Thank you, John. Again, please submit your questions via chat for discussion at the end of the webinar. Our next speaker is Dr. Toscano.
Thanks, Donald. So I'm going to give you a background on the regulatory guidelines from a non-clinical reviewer perspective for assessing ototoxicity in the development of both otic drugs and non-otic drugs. So before I start, I wanted to begin with this quote, which was from a paper about 5 years ago. And it states that there are currently no systemic regulatory requirements for any standard preclinical studies conducted of new molecular entities that include analysis of all peripheral and central vestibular structures or auditory sensory organs before drug approval. And this is just about as true today as it was 5 years ago. It's definitely generally a fair statement. However, in the cases in which a drug is being developed to be put into the otic space, we there is a requirement for more specialized assessments in the non-clinical studies. However, there are some measures in general tox studies that could be leveraged to assess potential ototoxicity of drug compounds that are being developed as non-otic treatments. So as far as the non-clinical safety evaluation expectations for otic drugs, that is, drugs that are being designed to be put into the otic space, so therefore the outer, middle, or inner ear, there is a guidance, a FDA guidance, and it's not specifically entitled as a otic drug guidance. It is the reformulation and alternative route guidance. And within that guidance, there are some recommendations for the types of non-clinical studies that should be conducted for the development of otic drugs, as well as other routes of administration. And I should start off before I go into the types of non-clinical safety assessments that are recommended. I should state that what I'm going to state here is all dependent upon you having a discussion with the appropriate review division that you're going to submit your application to, because that division may require other assessments depending on the nature of your drug product. So within the guidance, it states that if the ability of the drug product should be determined for the ability to penetrate the tympanic membrane and get into the middle or inner ear. If it is determined that this is going to make it into the middle or inner ears, or if it is specifically being designed to be put into the middle or inner ear via transtympanic injection, then the non-clinical studies that should be conducted on this should include a assessment of audition in the form of auditory brainstem response, as well as microscopy of all relevant locally exposed tissues, including a cytocochliogram. Now, you've heard the specifics of ABR and cytocochliogram in the two previous presentations. So what you'll notice right away is that there's actually something missing in this guidance, and that is the guidance is silent on the assessment of vestibular function and the structures that are related to vestibular function. Now, some things to consider when you are designing studies for assessing local toxicity testing of human otic drugs, again, drugs that are being designed specifically to put into the otic space. The route duration and formulation in the non-clinical studies should, as much as possible, mimic the clinical regimen. This is pretty standard for most types of drug products, especially route and duration. Formulation is much more important when it comes to the assessment, non-clinical assessment of otic drugs, because that testing the clinical formulation allows for an assessment of both the excipients, toxicity of the excipients, and the drug substance itself. The guidance that I just mentioned, the alternative route and reformulation guidance, is silent on the number of species necessary for the assessment of non-clinical. For non-otic drugs or drugs being taken through the enteral route, it's usually 2 species. That may be the case here. 2 species may be necessary. So therefore, adequate justification for selected species should be provided when you are submitting your application here. Many times we will see studies conducted in 1 species, and that would be just fine based on the justification provided. But as I said, the guidance itself is silent in the case of otic drugs. In other types of routes of administrations, it's very prescriptive. It's important to assess excipient toxicity when assessing, when performing non-clinical studies of human otic drugs, because the excipients that you may use in your drug formulation, in your drug product, may be in the inactive ingredient database, but for other routes of administration. That does not necessarily mean that those excipients would be without toxicity when put into the otic space. Therefore, inclusion of a saline control is recommended when conducting these types of studies, especially transtympanic studies where the drug is going to be put into the middle or inner ear, to allow for an assessment of the toxicity of the excipients. Positive control is also helpful. And primary pharmacology studies may also be helpful to provide a plausible biological effect to support studies for pediatric patients. As many of you know, there's a higher bar for testing drugs in pediatric populations, and that usually requires some sort of prospect of benefit, and that could potentially be justified by providing the results of primary pharmacology studies to demonstrate some efficacy of the drug product in animal models of disease. And as with most tox studies, it's always important to make sure that your high dose is justified. It should also allow for flexibility in clinical development and providing adequate safety margins. And in this case, those safety margins would be calculated on dose per perilymph volume. Because it is rare or almost never seen to have sampling of the perilymph from human subjects to determine what the exposure was, although we do see that on a regular basis in the animal studies. So calculation of dose per perilymph volume would allow for interspecies dose extrapolation. Some justifications that can be used for the high dose, one of them is maximum feasible dose. If maximum feasible dose is being used as a justification, it's recommended that a justification is provided in that it is explained why this is a maximum feasible dose. For example, it's higher dosing would be limited by dosing volume or drug concentration, et cetera. Middle ear tissue assessment is just as important as the ABR and cytocochliogram that you heard about previously, because it allows us to potentially determine if there is any hearing loss in the non-clinical studies, if it is of a conductive or a sensory neural nature. Many times the hearing loss can be because some drug product was injected into the middle ear, therefore creating a temporary conductive hearing loss. This middle ear tissue assessment of the ossicles and the tympanic membrane would be important in helping to determine if that is the case. And your ABR and cytocochliogram should be adequately conducted in that it assesses an adequate frequency range in the entire length of organ of Corti. Now, in the case of otic drug device combinations, there are additional guidances that are available. And I'm listing a few here that are provided to me by my CDRH colleagues. And I would recommend that if you are involved in or considering the development of a drug device combination, that you reach out to CDRH to determine what additional guidances you may need to consult prior to determining what types of non-clinical studies you would need to conduct. So now I'm going to switch gears here and go into the development of non-otic drugs. So basically everything that you don't put in your ear. In the case of those types of studies, we usually don't get a lot of information about the status of the middle and inner ear and hearing function. So are there measures in standard non-clinical assessments that are conducted in those cases that we could leverage to assess ototoxicity? And pharmacokinetic distribution studies, it's uncommon that we get inner ear and perilymph PK studies in non-otic drug assays or studies. But we do see distribution studies to the brain and CSF. We may be able to leverage those. Safety pharmacology studies could be used, but generally these are usually single dose studies, so they may not be as useful in assessing an ototoxicity that might be dependent on reaching steady state or multiple dosing. in order to manifest the ototoxicity. And also these use a very rudimentary assessment of hearing, such as a click and auditory response. Clinical signs can also be used, circling, head tilt, those types of things, but these are also non-specific for ototoxicity and also have low sensitivity. Also, auditory startle is a method that is received usually later on in development as a part of the pre-postnatal development studies or juvenile animal studies. So it's usually not available for first-in-human studies, so it would not be helpful in assessing ototoxicity at the earlier stages of development. So what can PK distribution studies tell us about inner ear exposure? Well, generally the inner ear is not assessed, as I stated, in PK distribution studies, but the brain usually is, and sometimes the CSF. Now, there is a connection between the inner ear and CSF via the cochlear aqueduct that connects to the subarachnoid space and the scala tympani. So there's a plausible CSF to perilymph connection. We do have some data from Alex Salt's lab that demonstrates that there is some flow rate of CSF into perilymph. Therefore, it's reasonable to conclude that drugs demonstrating distribution into the brain in PK studies, it is plausible that these have the potential to distribute to the perilymph via the CSF. So if we see it in the brain, it's possible it's also getting into the ear. We may want to make sure we keep an eye on this particular drug candidate to see if there are any effects that we can pick up that might be affecting the inner ear. Now, as far as auditory function assessments in routine non-clinical studies, like I said, these are generally non-specific and low sensitivity, and that is due to things or situations such as rodents can lose high frequency hearing and still respond to lower frequency noises that are used in common neurology assessments. Usually for auditory startle, these are broad spectrum noises such as white noise. So a rodent could definitely still respond to that sound, even if it lost a certain frequency band of its hearing due to an ototoxicity liability of the drug that is being developed. And also auditory startle consists of a motor as well as an auditory component. So it's not really specific to auditory dysfunction. So if your drug candidate is actually affecting motor activity, it could be misinterpreted as having a ototoxicity liability if you see something in the auditory startle assessment. Now, auditory startle can be modified to detect hearing loss, and there's a modification known as the reflex modification of the auditory startle, and this acts almost like a pre-pulse inhibition, where you provide a lower volume pre-pulse in a specific band of frequency that you're interested in prior to providing the louder white noise. And if the rodent is able to hear within that frequency, then the response to the louder white noise will be dampened. But if not, if there is some hearing loss within that frequency band, then there would be that response would not be dampened. So this could give you an idea of if there's a hearing loss within a certain band of frequency. But this would be sort of generally, you'd have to test it at multiple frequencies to be able to determine if there was some loss within a certain band of hearing frequency. As far as histopathology assessment of auditory tissues, the auditory tissues, the inner and middle ears, are not routinely assessed in general tox studies, except for the brain. STP, or the Society for Toxicological Pathology's recommended core battery of tissues that should be assessed in non-clinical safety studies, does not include auditory tissues. It interestingly includes tissues that are associated with all the other special senses, eye, skin, tongue, but it does not optic nerve, nasal cavity, but it interestingly does not include auditory related tissues. Now, as you heard earlier, assessment of auditory related tissues as far as histopathology methods are concerned, especially the sotocochleogram, are difficult to perform and tedious. So it's understandable that these are difficult to harvest and process. However, it's questionable if this is a sufficient reason to routinely ignore it in histopathology assessments, especially in light of how devastating auditory dysfunction and hearing loss can be in patients. So as far as future directions for non-clinical ototoxicity assessment, you heard some discussion of cochlear explants and cultured hair cells. These are good screening tools for doing initial work on drug candidates prior to animal studies. However, these are not as useful as in vivo studies and would not substitute for in vivo studies when determining safety margins. Also, consideration of possibly adding functional measures of hearing to a standard battery, say like a CNS safety pharmacology study where DPO-AE or something like that, or reflex modification of auditory startle could be added to those to provide a better readout for auditory function in general non-clinical studies. And possibly even development of sensitive, soluble, or imaging biomarkers of ototoxicity. HESI has a well-established program now where they are trying to develop a soluble or imaging biomarker of central nervous system neurotoxicity. So it would not be too much of a stretch to begin to try to determine similar biomarkers for ototoxicity if plausible. And finally, possibly an international guidance, development of an international guidance on ototoxicity testing. As I stated earlier, the alternative route and reformulation guidance from the FDA is really the only guidance out there on on conducting assessment of ototoxicity in non-clinical studies. So it would be interesting if this could be, if a discussion could be initiated at the international level on what type of guidance could also be provided for assessing ototoxicity.
All right. Thank you, Chris. In summary, this webinar has covered why it is important to screen for ototoxicity, some of the animal models and techniques used to assess ototoxicity, and the regulatory landscape for ototoxicity and to advance auditory therapies. This concludes this presentation. The speakers are now available for your questions in a discussion panel format. Thank you again.